Good morning, everyone. I'm glad that I got this opportunity to talk in front of all you eminent scientists. I'm a postdoc fellow with Dr. De Jesus Perez lab. To begin, let me share my screen. Um, yeah, I will be talking about HIV and methamphetamine use in relation to pulmonary hypertension, but I would like to take you through the journey. So before joining Dr. De Jesus Perez lab, I was working with Dr. Navneet Dhillon in University of Kansas Medical Center, where we were working on HIV-related pulmonary arterial hypertension in relation to drugs of abuse, which included cocaine and morphine. To begin with, let's so the if we talk about the epidemiology of HIV, IVDU, PAH, including, so IVDU is intravenous drug use. And if you talk about intravenous drug use, which includes morphine, cocaine, and methamphetamine and other drugs of abuse, HIV-PAH occurs in almost like 15% of HIV-infected patients, regardless of the route of infection. And if you talk about IVDU, it is a risk factor in almost 50% of PH diagnosed individuals, like who can get HIV, and almost 60 to 80% of HIV infected patients which are diagnosed with HIV with PH are intravenous drug users. So looking at these statistics, it becomes pretty evident that intravenous drug use along with HIV infection plays a critical role in development of pulmonary arterial hypertension, or vice versa, where HIV infected patients coming up with pulmonary arterial hypertension disease are mostly drug users, and like methamphetamine or PH, or other drugs of abuse like morphine and cocaine. Now, as Dr. Zamanian has already mentioned, that we are looking for for a double hit hypothesis for development of pulmonary arterial hypertension in these cases, where either we are talking about genetic predisposition, for example, in pulmonary arterial hypertension, we talk about BMPR2 mutations, along with some second hit like of a drug of abuse like methamphetamine or morphine or cocaine. Here, we kind of extrapolate this same hypothesis and think that HIV itself, infection itself, acts as a first hit to develop pulmonary arterial hypertension. And when there is a second hit of a drug of abuse, then the things become worse, and it leads to severe pulmonary hypertension. The mechanism that we talk here is that when these two hits like HIV infection and drug of abuse target these vascular cells, which are mainly endothelial cells and smooth muscle cells, it accompanies a lot of vascular remodeling, and also it accompanies a lot of inflammation around the vessels. So the idea here, when we are talking in context of HIV infection, is also that though it is known that the drugs of abuse like morphine, cocaine, or methamphetamine can directly interact with endothelial cells and smooth muscle cells, there have been no reports as such suggesting that HIV itself can interact with endothelial cells or the vascular cells directly. So usually the idea is that the viral proteins come from the inflammatory cells that are surrounding the vessels, and that's how this interaction happens, and which causes the further injury to the vessels. So so we are talking about here when the first injury that happens is on the endothelial cells, when there is loss of tight junctions on the endothelial cell layer, there is cell death due to apoptosis, and then slowly and slowly, what happens is that the some of the apoptosis undergoing endothelial cells become apoptosis resistant, and then the proliferation of those apoptosis resistant endothelial cells happen, and which also is accompanied by smooth muscle cell proliferation, vascular thickening, and leading to finally formation of plexiform lesions or to severe occlusion of pulmonary vessels. So going forward, here I'm showing a picture a panel of staining on HIV IV drug users lung sections, where you can see that both intravenous drug use and HIV independently can cause a lot of pulmonary vascular remodeling, where you can see the thickening of vessels. But when there is a both use of both HIV infection and combination of intravenous drug use, the things become pretty severe, and we can see a lot of smooth muscle thickening, occlusion, and also formation of plexiform lesions. So after this, since I have mentioned that we were working on both morphine and cocaine, Dr. Dhillon, before I joined, they have worked on SIV-infected morphine injected macaques. And here there is a panel showing severe pulmonary remodeling, and you can see plexiform lesion formation in SIV infected macaques that were injected with morphine. On the other panel here, I am showing a vessel from HIV transgenic rats. This is a rat model that is a transgenic HIV rat model created by Dr. Reed's group, and this is a rat model which expresses 7 out of 9 genes of HIV genome, and the GAG and polymerase genes are deleted, but it expresses all the other HIV proteins like TAT, GFP, and NAF, GFP120 and NAF. So you can see here and appreciate that there is a severe vascular remodeling and a lot of smooth muscle thickening that you can see, which has led to almost an occlusion of the vessel in these rats. Also, here the lower panel, which I'm showing, this is showing the tunnel staining in morphine or SIV infected or SIV infected and morphine injected macaques. And you can see that the pink color shows here the tunnel positivity around the vessels, which you can see that it is very much increased in the macaques that are injected with morphine and are infected with SIV. So this kind of led to the basis of the fact that when HIV or SIV or drugs of abuse like morphine and cocaine are given together, they synergistically act to cause endothelial dysfunction and also smooth muscle proliferation. I will not focus much on the cocaine injected rats or cocaine studies. When I joined this lab, my project was more on morphine related, morphine and HIV related studies, and I studied these on rats, HIV transgenic rats, and also on endothelial cells, focusing mainly on how morphine and HIV together leads to dysfunction in the pulmonary microvascular endothelial cells. So here will be moving more towards the basic research part, where we saw that when pulmonary microvascular endothelial cells, they are treated with morphine and HIV-TAT protein, we observed that the microvascular endothelial cells, they have a lot of ROS generation, reactive oxygen species generation, which you can see here as the morphine and TAT treated cells had increased ROS. And we were also trying to figure out what kind of ROS generation is this. either is it a mitochondrial ROS generation, or is it from general, or is it NOx related oxidative injury, or peroxisomal associated oxidative injury. And what we found out was that these cells have NOx related oxidative stress injury, and that's why when we treated with VAS3947, which is a NOx inhibitor, there was a decrease in ROS generation. Also, we observed that in the similar conditions, there is a lot of cell apoptosis, which was done through caspase-3 glow assay, and similar results were obtained where morphine and treatment showed significant increase in cell death, and which was decreased significantly when we treated the cells with NOx inhibitor VAS3947. The previous publications from this lab have also shown that morphine and TAT treated cells undergo autophagy, and there is increased levels of LC3B protein, which is a marker of autophagy in morphine and TAT treated pulmonary microvascular endothelial cells. And here we can see that the morphine and TAT treated cells have increased LC3B, and as you can see here in the staining of LC3B markers, and which gets inhibited by which gets inhibited when we treat with both VAS3947. And so this shows that probably the mechanism that is kind of playing a role in causing HIV plus morphine related endothelial dysfunction is through ROS mechanisms, where the cells undergo a lot of oxidative stress and then trigger the functions where first they undergo cell death and later they undergo maybe proliferation or lead to smooth muscle cell remodeling. I would like to also show you this data where we saw that HIV transgenic rats that were injected with morphine for almost 21 days, they have increased right ventricle systolic pressure as determined by catheterization surgeries. And you can see here that morphine independently also significantly increases the right ventricle systolic pressure, but when combined with HIV, the reactions are synergistic and systolic pressures are much higher. Similarly, the right ventricle hypertrophy also is significantly increased in HIV transgenic rats that were injected with morphine. Here, this is a panel showing VWF and smooth muscle actin staining on a small arteriole of HIV morphine transgenic rats. And you can appreciate that there was significant smooth muscle remodeling, and you can see a kind of vascular occlusion where both endothelial cells and smooth muscle cells have kind of intermixed, and there is a lot of endothelial proliferation, and it has become like a occluded vessel, which is almost becoming like a lesion. So, this suggests that definitely there is an indication that these drugs of abuse are very much prominent in causing pulmonary vascular remodeling. Also, the isolated cells from these rats showed increased ROS, and also when in vitro these isolated endothelial cells were treated with either NOx inhibitor or independently showed cell death. But in the animal models, we showed we got a different result in terms of that the cell death was kind of decreasing because probably it surpassed that endothelial cell apoptosis stage and went through proliferation. And also you can see here that these cells, as I told you in the ROS measurement, which was done fluorimetrically, here by staining also you can see a lot of cells that were DHE positive, which is a marker of oxidative stress. So, so after doing this, when I joined Dr. Vinicio's lab, here we are working on role of CES1 in meth-induced pulmonary endothelial injury. And
Stuti, could I ask you a question before you move
yes.
into this next section? Can you go back to there? So like a couple slides here, one was on the human cells that you exposed to the substance in HIV, and then the other is from the rats. One of the questions I have is, you know, you see very elegant remodeling of the arteries here. Is that process irreversible? So like, let's say somebody stops using this drug, or someone starts taking antiretrovirals. You know, what is the process that happens from there?
Yeah, so one of the things that we wanted to study was that when these rats are given naloxone to reverse the morphine effects, whether they would be able to reverse the pulmonary hypertension. But that study was not done. We also kind of were thinking that if we give antioxidants, maybe the things are reversed. But what I feel is that if the morphine injections are not chronic, because we were giving 21 days of injections every day, so the rats were already very fragile, and they were not, especially HIV transgenic rats. So there was lot of mortality also, and they were not able to. A lot of the rats were not able to survive the whole ordeal of giving for 21 days of morphine injections. So I feel that if we had given lower doses of morphine and then tried to keep the rats for longer time by giving not antiretrovirals, but to reverse the morphine injection, because I think morphine was creating a significant hit on the HIV transgenic rats. So that could have been reversible, but with chronic injections, I don't think so that it would be reversible.
Can I also add, from the clinical side, it's irreversible. It's irreversible. So once you get it, you keep it. And it's progressive. So for the HIV patients, the antiretroviral therapy makes no difference in regards to controlling the disease progression. Okay, so the incidence and prevalence has remained unchanged even on the wake of antiretroviral therapy, which makes this one of the most devastating complications of the disease. And obviously, we see it with med as well. You stop the med, but the patient continues to progress, eventually may need a transplant or dies. So we're dealing with complications of HIV and methamphetamine that are irreversible. Now, if you put the two together, you can imagine things may be worse. We don't know, but I think, again, in thinking of our blue skies, that may be an area of exploration. Thank you, Stuti.
Yeah, so continuing to the work that I'm doing here. So we actually are working on meth-induced pulmonary endothelial injury, because you have heard before from Dr. Zamanian and also from Dr. Vinicio that meth-induced pulmonary arterial hypertension is a disease which they are seeing in the clinic, and a lot of people are coming up with pulmonary arterial hypertension and who abuse methamphetamine. So this disease is of critical importance, irrespective of whether we are talking it in relation to HIV or in terms of idiopathic or APH. So the idea behind this study, where we are talking about role of SES1, which is carboxyl esterase 1 enzyme, is also somewhere related to the oxidative stress. And I would like to talk briefly about the carboxyl esterase 1 enzyme. So this enzyme is located in the endoplasmic reticulum and is responsible for the metabolism of toxins and different drugs. So what we have found in our earlier studies is that when these BMPR2 mutated patients which have lower expression of SES1 in their endothelial cells. So, so that kind of led us to think that this enzyme may be playing some role in metabolism of toxins and maybe playing some role in causing the endothelial dysfunction, which is related to pulmonary hypertension. And more so that, so this enzyme actually metabolizes esters. But if you talk about methamphetamine, it doesn't really give out esters as a first part of metabolism. But later on, if there is metabolic products of methamphetamine, which may release esters and which may be metabolized by SES1. So that was the idea of doing this study. And here I can show, here these are the slides, these are previously published data from Dr. De Jesus Perez lab, which shows that in meth-PH patients, you can see here that there is high expression of SES1, but in cases of meth-PH human lung sections, there is very less expression of SES1 enzyme. And also the SES1 activity that was done in PBMCs of meth-PH patients is very less as compared, significantly less as compared to the patients that were controls. So moving forward with the idea that probably here also the oxidative stress mechanisms may be playing role, we kind of went ahead to see the effect of methamphetamine treatment on pulmonary microvascular endothelial cells in terms of ROS generation. And we found that with increasing concentrations of methamphetamine till 500 micromolar, there is a significant increase of ROS generation. Further, we also kind of we verified this these observations by doing staining for ROS, and here you can see that both cell ROS in general and mitochondrial ROS that was done by MitoSox assay, we can find that we saw that in SES1 knockout cells we found which were exposed to meth, there was increased cell ROS and also mitochondrial oxidative stress. Along with that, we also went ahead and saw that what is the effect of methamphetamine and SES1 knockdown in microvascular endothelial cells in terms of autophagy and in terms of ER stress proteins, which are CHOP. And we found that there is increase in both LC3B and also the CHOP protein, suggesting that there is some endoplasmic reticulum stress that is also happening in these cells. These studies are still being done, and I am extrapolating these studies further to study whether methamphetamine and downregulation of SES1 contributes to metabolic dysfunction in terms of lipid accumulation and also in terms of mitochondrial dysfunction and ER dysfunction. So moving forward moving forward with functional assays, we also found that these, when we expose SES1 knockdown cells which are exposed to meth, there is increased cell death and which is rescued when we treat the cells with plasmids which are overexpressing SES1. So this shows on one hand that maybe this is another mechanism where definitely oxidative stress is playing a role, but the enzyme carboxyl esterase 1 is playing a critical role in maintaining endothelial cell function. Now moving forward to the relation of meth and TAT proteins or HIV in endothelial cell death and also the SES1 expression. So when we were thinking about all this, we also kind of thought that maybe similar to just like BMPR2 mutation, maybe, or even in idiopathic PH, if SES1 is playing a role in causing, in promoting endothelial function, or meth is causing a lot of oxidative stress and endothelial cell death, maybe if we give HIV-Tat protein to these cells with or without exposure of meth, maybe we will see similar kind of response. And indeed, we saw that even though meth alone also produced a lot of ROS, the amount of ROS was almost synergistic when we did treat the cells with HIV-Tat protein along with methamphetamine. Also here, you can see that the in vitro, when the cells were treated with both meth and TAT, there was a significant decrease in the expression of SES1. And moving further, we also showed that there is significant synergistic cell death when we treat the pulmonary vascular endothelial cells with both methamphetamine and HIV-Tat protein, and which was also much more significantly higher when we treated meth and Tat in presence of when the cells were knocked down with SES1. So this all studies show a significant correlation between what mechanisms may be playing a role in either HIV meth or just BMPR2 downregulated meth exposed cells, or maybe just in meth IPH or meth exposed pulmonary arterial hypertension cases. And this kind of is laying a very good background of saying that maybe oxidative stress is the mechanism that may link all of this together. So what we propose here as a model is that when the meth and viral proteins enter inside endothelial cells, they increase ROS, which leads to mitochondrial and ER damage, which finally causes endothelial apoptosis and vascular injury. Whereas in healthy, there is SES1, and there, and also even if the people are, and so the mitochondria is healthy and there is no ROS, and hence there is the endothelial cells work fine and there is vascular repair occurring. So with this, I would like to acknowledge both the labs where I did this work. First of all, Dr. Vinicio de Jesus Perez lab and the members of the lab, and Stanford University, and the next Dr. Navneet Dhillon and lab members in University of Kansas Medical Center. Thank you.
Thanks. Questions?